Hi, welcome back to GCB 531. I'm John Murray, Assistant Professor of Genetics here at Penn, and today's lecture is going to be about genomic inheritance, or to use the old-fashioned name, genetics. It's going to be the first of a series of 3 lectures on genetics and genetics and how genetics applies to our thinking about genomic data sets and some of the next generation sequencing as we move into the next 2 lectures. But today we're going to talk really about basic genetic principles, and then in the next 2 lectures we'll talk about experimental genomics and genetics and clinical genetics. So this is an outline of what we'll talk about today. So first you might wonder, well, why do we need to even think about sort of traditional kind of genetics in the genomic era? So I'll give a brief summary of why I think the genetic principles are still critical to understanding how genome functions in disease and in other aspects of biology. And we'll go through some sort of basic Mendelian mechanisms and how genes are inherited across generations. This is not going to be a full course on genetics, since we're assuming that you have a certain amount of background in genetics. And so if this seems really fast, and it might be worth going back and looking at some of these slides again. There's no shame in that, especially if you don't have a really strong genetics background coming in. Then we'll move on to some non-Mendelian inheritance. So that's more complex patterns of inheritance. And finally, we'll sort of synthesize this by looking at how these different forces act to shape the structure of the genome in populations. All right, so why is genetics important in this genomic era? So we can start with a definition of what is genetics. So this is just a quote that I got from Wiktionary.com, our one of our favorite open source dictionary. It's a branch of biology dealing with the transmission and variation of inherited characteristics, in particular chromosomes and DNA. Okay, so that's a reasonably good definition. Sort of the key aspects there are these inherited characteristics. The term we use for those is phenotype. So that would be something like, say, eye color, hair color, whether you have cystic fibrosis or not, et cetera, et cetera. Then there's the genotype, and that's actually the underlying DNA sequences that control your phenotype. So in total, your phenotype is really the sum of all of the different characters that you have that are controlled by your genetics. And then the term gene is a little bit tricky to define. It sort of evolved as we learned more about the molecular structure of protein coding genes in the genome. But in this lecture, we're thinking about genes really as individually separable DNA sequences that encode for particular phenotypes. And that's a little bit distinct from the traditional molecular biology definition of a gene, which is really meant to represent a particular mRNA that encodes, for example, for a protein or perhaps a non-coding RNA. And so there are definitely cases where a single transcribed sequence that actually gives rise to different phenotypes that are genetically separable and distinct depending on which parts of the protein, for example, are mutated. And there's also examples of the reverse. All right, so one of the really critical, as an experimental biologist and as a systems biologist, reasons to continue thinking about genetics is that genetic principles really tell us about the causality of biological phenomena. And so you can do all sorts of high-throughput experiments to look at, for example, which proteins interact with each other. You can find all the genes that are expressed in one tissue or another tissue and find genes that are co-expressed in those. But it doesn't really tell you that those genes are actually working together in the same process. And what the gold standard is to figure out that a gene is really important for a particular process is its mutant phenotype. So that's what is the phenotype of the organism when that gene is altered in some way. And if you have a number of genes that have similar mutant phenotypes, that actually is a really powerful predictor that those are actually working together in some common process. And so when you combine it with these other types of large-scale data, it really ties things together. Okay, so now I'm an experimental genomicist, but I think that's going to be a really tiny fraction of where genetics touches on people's lives in society. And one of the big things that's really occurred over the last 10 years or so is the advent of personal genomics, and in particular direct-to-consumer genomics. So companies like 23andMe, for example, which you can pay them a few $100s and they'll give you back this really high-resolution map of what your particular genotype is, which different variants you have at different places across your genome, and tie that into your ancestry, your risk of different diseases, and so on. And so really to understand these kinds of information, you really need to understand the genetics. And I should mention that this is more and more being used in the clinic as well. So it's not just in these personal direct-to-consumer kind of approaches. Okay, and then so a third case where genetics is really critical is in cancer, and we'll talk about this a little bit in the clinical genomics lecture. But the key thing to remember is really cancer is really a genetic disease acting at the level of single cells. And so if you have a single cell, really it's going to be if it has a particular mutation, for example, that inactivates a particular tumor suppressor gene, that causes the cells then to proliferate, and you get this sort of initial clump of hyperproliferative cells, and then you'll have additional mutations. And so the same processes of mutation and selection that we'll talk about later in this lecture apply on those cells individually and cause them to overgrow and eventually become cancer in some cases. Okay, so Mendelian inheritance. Now Mendel who was famous for originally coming up with some of these ideas, and really developed the first description of genetics looking at peas, right? And so famously, he crossed yellow peas with green peas, and you can sort of denote the relationship between genotype and phenotype using these capital and lowercase letters. So everybody has, if you're a diploid, which all humans are, and most model organisms are as well, have 2 copies of pretty much every gene. And if you have 2 different alleles, so these letters are called alleles, you might have 2 of the same allele, in this case little y, and that means the piece will be green. Or you might have, say, 2 different ones, maybe this big Y and little y. So that's two different alleles, and depending on which of those alleles is dominant, you'll be either yellow or green. And so in this case, the yellow allele is dominant, and that means that if you have one copy of that, it doesn't matter if the second one is big Y or little y, you're going to be yellow peas. And this is the simplest way to follow these is through one of these Punnett squares, where you have if you have a cross between, for example, this little y, and this big Y p, then each of the progeny is going to get one allele from each parent. And that, as long as, in most cases, is going to be a random choice for each parent. And so you'll get either a little y or a big Y from this parent, and you'll always get a little y from this parent. And then you can just look at the ratios of the progeny, so you'll have 1/4 in each of these boxes. And the result of that is you end up in this particular cross, you get half yellow peas and half green peas out. And so by following the patterns of inheritance, you can really get a sense of which genes are involved, or how many genes are involved in this particular character. And the key thing to remember is that these are not really abstract things. Big Y and little y are actually individual DNA sequences on the chromosome. And so, in this case, the big Y, little y heterozygote, you have one chromosome has a big Y, which is some sequence, and the other chromosome has a little y, which is some different sequence. So just a little bit more about chromosomes and how the DNA sequence is actually related to these alleles. As a reminder that in the case of humans, we have a total of 46 chromosomes, 2 copies of all of the autosomes. That's chromosomes 1 through 22. And then if you're a male, you have an X and a Y chromosome. And if you're a female, you have 2 X chromosomes. And just to reiterate, then each individual nucleotide along these chromosomes could be a potential genetic allele if it causes some phenotype. And so we can think about the transmission of this big A and little a allele, for example, which are on chromosome 11. Now, again, if you have 2 heterozygotes for big A and little a, their progeny, you're going to get randomly either a big A or a little a chromosome from each parent, and you end up, again, with 1/4 getting 2 big A's, 1/4 getting 2 little a's, and 1/2 getting 1 copy of each type. And this, these alleles, they could be any variety of different types of sequence changes. So, for example, I just showed a small region of a chromosome around the hemoglobin gene, which happens to be on chromosome. This is the beta globin gene, which happens to be on chromosome 11, and mutations in beta globin cause a number of diseases, for example, sickle cell anemia, beta thalassemia, to name 2. And this is just showing that this is the actual gene structure of hemoglobin or beta globin, and there's a number of individual sequence changes that are common in the population. So all of those could be alleles in addition to these disease mutations. All right, so one key aspect of inheritance, now is if 2 genes are on the same chromosome, you're not going to get random assortment of those 2 genes. Instead, they'll tend to go together with each other. And so if you have now this A plus and B minus allele on one chromosome, A minus and B plus allele on the other chromosome, the more than you expect by chance, you're going to get, say, A plus and B minus going together, or A minus and B plus going together, and so you'll get co-segregation of those syntenic alleles. However, there is recombination, and this has been talked about in previous lectures, so you do in fact get assortment of genes even if they are on the same chromosome. And we can take advantage of the frequency of that recombination to actually map genes to particular spots on the chromosome. So here's a question on section 1. So you're studying a newly identified species of Martian. One important thing about genetics is that it can be any species with Mendelian genetics. The same principles apply. And these Martians have either red or purple eyes, controlled by a big P gene. So you have big P, that's dominant, and you have purple eyes. And if you have a little p, and you have two of that alleles, then you have red eyes. And so which of the following crosses will produce 75% purple eyes? So the answer is that this cross between the two heterozygotes is going to give you 75% purple, because big P, which is purple, is dominant. Then if you get, from this cross, if you get either 2 copies of the big P gene or 1 copy of the big P gene, you're going to be purple. And so in the Punnett square, that covers 3/4 of the possible outcomes. So what types of sequence changes do we see in the genome? So I wanted to summarize before we actually start talking more about how different mutation or different sequence changes give rise to different phenotypes. We talked in one of the earlier lectures about copy number variations and structural rearrangements. So you can get deletions. In this case, it's a fairly large deletion of a part of a chromosome. You can get duplications, where you can also get inversions. You can get translocations. But then there's also, and these actually, even though there are fairly small numbers of these in any given person's genome, because they're relatively large, they cover a fairly large amount of genomic sequence. And then the bulk of the individual mutations are actually going to be either single nucleotide polymorphisms, where, for example, you convert, in this case, this T becomes a G, so that's just a single base change, or small insertion deletions. Here you have for example, 2 bases that are deleted in this particular sequence. Okay, so I've been trying to be careful to use the term variant as opposed to mutation. So what is the difference between a mutation and a variant or a polymorphism? So the definition of a mutation is a deviation from the normal DNA sequence for a given species. But all species carry lots of sequence polymorphisms, and these are all of different rates really in the population, but they can be very common, they can be very old, they can even cause phenotypes. So really what you call a mutant, it really depends on what is the definition of normal. And so I think most people would agree that sequence changes that give rise to severe diseases, for example, cystic fibrosis or sickle cell anemia, for example, those would be mutations. But is red hair a mutation? And there's all these examples of traits that are strongly genetic, and it's not, either they're clearly not bad things to have in the population from our perspective, or maybe there's other cases where it's maybe vague. So like there's some idea that maybe different types of intelligence may predispose to different kinds of psychiatric conditions, for example. And if you get rid of all the risk of those conditions, you also get rid of particular beneficial classes of intelligence in the population. So an alternative definition is to use allele frequency. And so here the idea is just you're forgetting about whether the mutation is good or bad. If it's common, more than 1%, then we'll just call it a polymorphism. And if it's less than 1%, then we'll call it a mutation, even if it's neutral. And that, even though it's also imperfect, it's at least a fairly quantitative and reproducible definition. So there's lots of different ways that mutations can alter protein function. And most genes correspond to protein encoding parts of the genome that are transcribed and translated into protein. And you could have single nucleotide polymorphisms that cause changes in the protein sequence, for example, missense, where you have just one amino acid substituted for another, or nonsense, where you actually truncate the protein by having a stop codon where you should have had an amino acid. You could have a small insertion deletion, or this also can occur with splice site mutations, where you insert a number of bases that's not a multiple of 3, and that will cause you a frame shift. So basically, all of the protein sequence downstream of that will be disrupted. And then there's a whole class of mutations that don't explicitly act on the coding sequence, but can still have severe effects. For example, regulatory mutations. If you get rid of an enhancer or promoter sequence that's important, you might not express the gene in the right places anymore. Splice site mutation could be, for example, you could now take a gene that's supposed to be spliced one way and cause it to be spliced differently, or you could alter the ratio of alternative splicing. You could have a mutation that causes the RNA to become unstable or too stable. And then you could have, in some of the larger rearrangements, you can actually have deletions of whole genes. You can, in an inversion or a translocation, you can actually create novel genes where you have part of one gene fused to another part of another gene, and that can cause detrimental effects. Right. And so these different types of functions, we sort of grossly group them into 2 categories: loss of function and gain of function. And loss of function, generally, if you get rid of a protein, that would be a loss of function. Usually those are inherited recessively, but there's cases where that's not true. One of the most famous is the RB gene, which predisposes to retinoblastoma, which is a kind of eye cancer. And if you just get one bad copy of that, that's enough to guarantee that you're going to get that cancer. Just because it turns out that you're guaranteed to lose the other copy by some kind of mutation mechanism in some of the cells in the eye eventually. You can also have a gene where you really need to have both copies, and one copy isn't enough to maintain normal phenotype, and that also will cause dominant inheritance, even when you have a loss-of-function mutant. And then gain-of-function mutants. There's all sorts of different kinds. You can have just the normal function is amplified in a particular protein, or you could have actually the opposite, where you make a mutant protein that actually interferes with the normal protein. Maybe you have a protein that dimerizes, and so when you have a bad copy, that binds to the normal copy, and now both of them lose their function. And you can have these neomorphs. So one example of that is these fusions where you have completely new genes with parts of one gene fused to another gene, but you can also have similar kinds of things where you have mutations in a particular gene that causes it to do something that it wouldn't normally do. And those are typically, again, but not always, inherited in a dominant fashion. And so we've learned a lot over the years from studying Mendelian diseases, and really the lessons are pretty parallel from experimental genetics in model organisms, where you've done genetic screens and identified the mutations that are responsible for the phenotypes in those. And sort of the take-home messages from those, first of all, is that by and large, the mutations were not in genes that we would have been able to guess if we hadn't actually done the hard work of figuring out what was involved. So you can't just say, oh, well, this person has a particular disease, perhaps it's this gene, and that just doesn't work most of the time. Most of the severe sort of high effect mutations, which was what you see in Mendelian disease, the single gene Mendelian diseases, affect the proteins, and so roughly half are missense, and a lot of the remaining are either nonsense, splice site, or either, severely affect the stability of the RNA or the structure of the RNA. And so actually it turns out that in this category of mutations anyway, it's rare to see regulatory mutations especially regulatory mutations. But that may not be true for more common diseases. So I just wanted to put up some examples of some Mendelian traits and look at how they're inherited. This pedigree got a little bit shifted. Some of the classic examples of recessive traits are cystic fibrosis. These are mutations in the cystic fibrosis transmembrane receptor, which in humans the major phenotype is in the lungs. Phenylketonuria, which is an inability to properly metabolize phenylalanine. These people have to go on a severely restricted diet. Sickle cell anemia, albinism. So these are all fairly severe diseases, and the sort of characteristic of recessive traits is that that you have about in at-risk mating. So here's two people who are having a number of children for this, and because they're both carriers for this, now 1/4 of their progeny are expected to be affected on average. So you don't tend to get vertical transmission. Whereas in contrast with dominant traits, some of the classic examples are Huntington disease, achondroplasia, which is dwarfism, Marfan syndrome, which is a fibrillin mutation. These are a characteristic set of phenotypes. Now, in this case, remember, it's the big A allele is the one that's causing the disease since it's dominant, so you see that disease actually vertically transmitted within families, and so it's passed on from parent to child, and you see this vertical transmission. Okay. And then there's a third class. We talked briefly about the sex chromosomes. Remember, males only have one X chromosome and a Y chromosome, and so the genes that are on the X chromosome have a particular mode of inheritance where they're much more common in males. So things like hemophilia A and red-green color blindness have that kind of characteristic. Okay, so mutations, they're not necessarily a bad thing. So otherwise, we wouldn't be nearly as interesting as we are today. So, some of the classic examples of fairly recent mutations in the human population that are arguably beneficial are lactase persistence. So there's a number of populations around the world which rely on dairy for a significant fraction of their calories, and so they domesticated animals to produce milk. And normally humans lose their capacity to digest lactose, the sugar in milk, fairly early in development. But so a number of populations have mutations that cause persistent expression of lactase later on in life and allow them to continue to digest dairy. Similarly, there's mutations in some populations that cause resistance to HIV infection. There was a recent paper a few weeks ago looking at mutations in a gene called APP that in those mutations make it so you're much less likely to get Alzheimer's disease, which I think everybody would agree is a good thing. Now, it's not to say that these mutations aren't likely to have other effects besides these. And so it really depends on what it may be that in the case of, for example, the APP mutation, the whatever defects that may result from that may be less severe than the obvious benefit of having a reduced risk of Alzheimer's disease in a modern society. And just briefly to mention that some recessive disease alleles are much more common than you would expect them to be based on how severe they are. For example, sickle cell disease and cystic fibrosis. And those are a category which the thinking is that there's something called heterozygote advantage. And so in the case of sickle cell, for example, heterozygotes don't have sickle cell disease, but they are resistant to infection by malaria. And similarly, there's a thinking that maybe cystic fibrosis heterozygotes might be resistant to cholera, and that those benefits may actually explain why those diseases are so common in certain populations. Okay, so if we have a particular trait that we think is genetic, and we'll talk more in the next lecture about how we actually know a trait is genetic, but say we want to know where it is in the genome, the classic way to do that is with linkage. And so remember, these genes are on chromosomes, and so you can imagine a particular disease gene, for example, is on chromosome 10, and there may also be a single nucleotide polymorphism on chromosome 10 nearby, where some people have, say, an A allele, and some people have a C at that position. That's just depending on which individual you are, you might have one or the other. It doesn't necessarily cause any phenotype, whether you have A or C, but it's a useful marker for that location in the genome. And so here we have, for a recessive trait, now a person who's homozygous for the recessive trait. So this person, we can call that it's recessive, so they're little d, and this person is a carrier, so they're big D, little d. And so the question is, which of their progeny, which ones are affected? And you can see that these people have 4 affected progeny, 4 unaffected progeny, which is what you expect by chance for this particular cross. And if you look carefully, all of the people that are affected also got the C allele from mom. And the people who are not affected, they all got the A allele from mom. And so that suggests, well, maybe there's some relationship. These things are co-segregating. And so we can do statistics on this to ask whether this is happening more often than you would expect by chance. And the way this is done classically is with something called a LOD score. This should be 3.6. So, and again, the point is not necessarily to be able to calculate this, but the concept is an important one. So the way you figure out basically the significance of the observed linkage is to count the number of recombinants and non-recombinants. So in the example we just looked at, we had all of these people, 8 non-recombinants, and we had 0 recombinants, because everybody got, basically, we have the C SNP with the disease, with the little d allele for the disease allele, and the A SNP with the big D allele, and that's how all of these people inherited that. So we have 8 non-recombinants and 0 recombinants, and so we can just compare how surprising would that be if these were tightly linked? So that's this top term. So that's just the probability of observing the pedigree that we had if they're linked with a particular distance. And we can try different distances. So we can say try 0.01, 0.05, 0.1, and try different possible distances, and then we compare that probability with the probability of getting the observed pedigree if they weren't linked. And so that's just going to be 1/2 to the number of informative individuals. And that ratio then, and the log of that ratio is the LOD score, and it becomes a fairly intuitive number basically. So if you have a LOD score of 3, that means that the pedigree observed is 1,000 times more likely if the gene is linked to the marker at a particular distance than if it's not linked. And if you can find the distance that gives the highest LOD score, and that's sort of the most likely distance between your marker and the disease allele. And this should be 3.6. So that basically, if you have a LOD score above that level for a traditional linkage study, that tells you that this is significant at the genome-wide level after you take into account multiple testing. Just throwing out there, we're not going to talk about it too much, but the phase is important. So in the case we showed before, you actually didn't know before you looked at the individuals whether the C allele was with the disease or with the normal. And if you know that, you actually get a little bit of a statistical boost, is all. So these types of this basic statistical framework is the same as what's used for much more sophisticated modern sort of association type studies, but with slightly different tweaks. And so that's why I thought it's important to mention it now. Okay, so here's a second question. You sequence the genome of an individual who has a dominant disease and identify a heterozygous missense mutation. It's in a protein kinase gene. And you test for this mutation in the other members of the family and find that all 23 of the affected family members and none of the 19 unaffected at-risk family members carry this missense mutation. So is the disease caused by the protein kinase mutation? Is the mutation significantly linked to the disease? Is it caused by a different gene near the protein kinase with the mutation? Or is the half of the normal protein kinase dose not sufficient for normal physiology? And the answer, best answer here is that the mutation is significantly linked to the disease. This is very, very significant. We have 42 non-recombinants and no recombinants. That's a really huge LOD score. It turns out it's more than 12. However, we haven't proven that the mutation we identified is actually causing the disease. It could also be that there's another change near our mutation that's linked to it that is the cause of the disease. And so since you have some whole genome sequence data, you may be able to find other candidates there or rule out other candidates. But really, to nail this down, the point I wanted to make here with this question is that linkage doesn't give you necessarily the exact gene by itself. You really have to do some kind of other experiment or logic to figure out which gene is actually important. So a lot of times what people will do is find, either in the mutant cells from the mutant individual, you can try and add the gene that you think is important back, or you can make a model in model organisms. So you can say add your mutation into a mouse and see if the mouse has similar phenotypes, or so forth. Okay, so the third section is going to be non-Mendelian inheritance. So Mendelian inheritance is all well and good, but it really isn't the rule when you look at the kind of human variation that we see in our day-to-day lives. And this is just a classic plot from early work by Francis Galton, well over 100 years ago, comparing the height of an individual with the height of their parents. And so you can just average the height of the parents, and it's true that if you have tall parents, you tend to have tall children. If you have short parents, you tend to have short children. But there's two big differences here from a Mendelian trait. One, you're going to have all different heights. There's not just tall and short. and two, there's this sort of you don't get this sort of simple Mendelian ratios where you have tall children always tall or short being dominant in the strict sense. Right, so you have a lot of the if it was Mendelian, you'd expect the medium parents should have plenty of really tall and plenty of really short parents, but instead you get these sort of bell-shaped curves around the mean. Right? And so there's lots of different ways that traits can deviate from Mendelian inheritance. One is that they may not even be fully genetic, so there can actually be stochastic or environmental aspects of a trait that interact with the genetic component. And we're not going to talk a lot about things like epigenetics, which is sort of, in some ways, in some cases, it's an environmental stimulus that causes it, but basically that's non-DNA sequence-based inherited changes, and there are examples of that. We're not going to talk about X-linked any more than we already have. And mitochondrial inheritance is another important sort of special non-Mendelian inheritance case where you have inheritance really just from mothers to all of their children, because that's how the mitochondria are passed down through the fertilized egg and not through the sperm. But we're really going to focus on complex traits, complex genetic traits, and mostly polygenic traits, and how those can give rise to more complicated patterns of inheritance. Okay, so here's a fairly simple example where you have now, instead of one gene, there's two genes. There's R and there's W, and these genes are really completely independent of each other. And so if you have two copies of little r or two copies of little w, you have this phenotype, which in this case is denoted by the white circles. And if you have big R and big W, then you have the red case. And now you get this, these more complicated ratios. In this case, you're 7/16 of the white circles and 9/16 of the red circles. So this is an example of locus heterogeneity. So basically, the same trait can be a result of multiple different genes. But these are, again, independent, basically acting individually as individual Mendelian kinds of genes. Now, genes don't have to be independent, though. A lot of times, genes that have related phenotypes actually are related to each other in some sort of a pathway. And when this happens, you can actually get much more complicated arrangements of the phenotypes when you start looking at combinations. So here's just sort of a toy pathway where you have gene A regulating B, C, D, and then ultimately a particular phenotype, and then a parallel pathway through E, F, G, and H, and then these pathways are cross-related to each other. So genetics, it turns out, is a really good way of figuring this kind of thing out. So in this case, there's all sorts of different types of mutations that could, in combination, act in sort of not the traditional Mendelian sense. And so, one classic case would be a synthetic lethal. So it may be that you really need both branches of this pathway, and if you knock out D, well, it's not such a big deal because you still have this pathway that goes through H. But if you knock out D and H, then you're in bad shape. You don't have either pathway, and maybe you're dead, or you have some strong phenotype. So this would be a synthetic lethal, or it might be an enhanced relationship would be another way if it's not actually lethal. Similarly, if let's see if I can get rid of these. So if you have a mutation for F, now you're going to have too much B. You're also going to have too much G, and maybe you're going to have a problem. But the problem is because you have too much D and H. But maybe if you knock out F and then you also knock out H, well, now you have too much D, but you don't have H anymore, and so you have sort of the right amount of activity, and you go back to normal. And so now H is a suppressor of F. And so you can actually work out from the double mutant phenotypes, in some cases, the order of a pathway like this. And you might hear the term epistasis, which I don't really like, sometimes used to summarize this logic. So here's just a Punnett square example of one of these polygenic cases where you have a synthetic phenotype. Now, we have the same cross that we had a couple of slides ago, where you have this RW double heterozygote But now instead of having 7/16 of the progeny having the white phenotypes, only 1/16 have the white phenotype, and the rest have the red. So so that's a synthetic example. But now we've still been talking about binary phenotypes, and we know, just like for height, and here's a nice example, for petal color, that you get not just one color and one height, but you have this whole range from this dark red all the way up to almost white petals, and this same kind of quantitative variation occurs for all sorts of traits. If we mentioned height, cholesterol, this can be a risk of diabetes. So actually be the risk of a nominally binary endpoint. So these traits, which now are continuously variable, we can call quantitative traits. Right. And so one sort of really simple way you can get something like a quantitative trait even from a single gene is if you have this incomplete dominance relationship. So now instead of having, here's a cross between big A, little a, and now if you have one copy of big A and one copy of little a, instead of being red, which would be for a traditional dominant relationship, you're sort of purplish pink. So you're sort of halfway in between. This would be incomplete dominance. And now you get this 1 to 2 to 1 relationship in the Punnett square, and you add some maybe blurring from environmental influence, and this actually can explain a lot of fairly broad quantitative divergence or divergence already. You start adding more and more genes together. It's very easy to make a really phenotypic continuum. So here with 3 genes now we've got all different populations, but it's really unlikely that you're going to get all of the white allele or all the red allele. More likely you're going to get some mix, and if you assume that the trait is just a result of the number of red alleles you have, then you start getting this bell-shaped curve out from where most of the individuals are going to have this intermediate phenotype, but there's going to be a range from low to high. Okay, and I mentioned that this doesn't have to apply to quantitative traits. This can also actually, risk itself can be the quantitative trait. And so you can have, one example was with risk of diabetes, right? So you either have diabetes or you don't have diabetes, but your risk can be a result of your underlying genetic constitution. And so, if you have too many of the bad alleles in this sort of simple model, then you get over the threshold, and then you're affected, or you have a high risk of being affected. So here's this an example with type 2 diabetes, where this you can measure both this underlying glycosylated hemoglobin, which is a trait, which is continuously variable, but then type 2 diabetes occurs when you're at the high end there. Okay, so just sort of to add one more wrinkle, the typical monogenic disorder, you have this simple genotype-phenotype relationship with environment and chance playing a fairly minor role. For typical common diseases, you have fairly large influence or fairly small influences from many different genes potentially on the phenotype, but then you also have many different environmental modifiers as well as chance potentially playing a role in sort of stochastically modifying the phenotype. Okay, so for the last bit of the lecture, I would like to talk about how these forces of mutation interact with natural selection and give rise to genome structure. Right, so sort of the key active force that's actually changing the genome when you start from a constant genome is mutation, right? So how does DNA sequence change arise, and for a reasonable sized population, by definition, any new DNA sequence change is going to be a mutation because it's going to be rare in the population. So the sort of two main mechanisms are by either errors, so the DNA polymerase is going along, replicating the DNA and makes a mistake, or maybe this can also happen in recombination. And if you fail to fix that, so the cells do have proofreading mechanisms to make sure that if they put the wrong thing in, that they take it out and put the right thing in, then you'll end up with a mutation. And then there's a second category, which is environmental, and this is why you want to avoid spending too much time in the sun. For example, if you have UV light will damage the DNA, especially in your skin cells. In the case of UV, it's by causing thymidine dimers to form in your DNA. And again, you have mechanisms to repair that damage, but with some frequency, you'll fail to repair it, and you'll have mutations there as well. And so when you look at the germline mutation rate, so that's what this this top number is. It's been estimated by a number of different ways, most recently by sequencing a bunch of small families or trios where you have both the genome sequences of the two parents and the child. It looks like the mutation rate is about 10 to the minus 8. So remember, the human genome is about 3 billion, 3 times 10 to the ninth base pairs, and it's diploid. So this corresponds to about 60 new polymorphisms per generation. And this is in addition to at lower frequency new structural variations, deletions, copy number variants, etc. And this sounds like a lot of mutations, but remember, most of the sequence is not encoding for genes. Only about 1% of the sequence, for example, is protein coding, and maybe about 5%, or possibly even less, is under strong purifying selection, which we'll get to. So really, this corresponds to about one mutation in a protein coding gene per generation. Okay, so mutation is just one of the features that actually contribute to diversity in the genome sequence. And so we talked about that new alleles arising at a particular rate. And once those alleles come along, they're subject to selection and to drift. And so selection, as an intuitive concept, makes a lot of sense. If you have a mutation that decreases the chance of survival for yourself or your progeny, that's going to be less likely to be fixed later in the population. That is, become more prevalent and eventually become the only sequence that's present. And if you have a mutation that increases the chance of survival and passing that gene on to your progeny in future generations, then that is more likely to increase in the population, to become fixed. And so we can define sort of as extremes, and normal fitness is 1, then a carrier, if you have, say, a dominant lethal mutation, that would have a fitness of 0, assuming it's lethal prior to reproduction. But then so that makes a lot of sense, but there's also drift, and drift is not quite as intuitive in some cases, and so alleles can just by random chance become more or less common in the population. And remember, we had those 60 new mutations. Maybe 55, maybe 59 of those mutations are going to be neutral or almost neutral. They're going to have little or no change in actual function. They're not going to impact phenotype in any way. And so those are just going to drift up and down by random chance, or if they happen to be lucky, then maybe they'll be next to a beneficial mutation and come along as passengers. So just as a demonstration of drift, one of the interesting features of drift is it is a function of population size. And so with a really small population, these are just some simulations. If you have 20 individuals in a fairly small number of generations, you start with equal allele frequencies. Many of those alleles will drift to fixation. You'll have 100% of one or the other, and not both, within a fairly small number of generations. Whereas with a very large population size, even over dozens of generations, you never get a very substantial change in the allele frequencies. So it turns out that the relative quantity for this is actually the effective population size, which is different from the real population size. So there's however many billion humans, but our effective population size is only 10,000 or so, it turns out. And that's because of historical bottlenecks in human population size. Basically, the effective population size is more related to the ancestral population size than it is to the current population size over short time frames. Right. So if we and this is from the theory of neutral evolution, and I'm putting some equations up here. The point is not the equations, but more the concepts. The important thing here is that the relationship, there's a fairly well understood relationship between this heterozygosity, which is given by theta, and that's just the fraction of individual nucleotides in the genome where any given individual is heterozygous. So how often, if I take one of my sequences and one of your sequences, how often is there going to be a difference? And for humans, that number is about 1 in 1,000 bases are different typically. And if you combine that with the mutation rate, that gives you actually this estimate for the effective population size of about 10,000. And that becomes useful for all sorts of other calculations as well. Okay, so yeah, it's worth mentioning that even though 1 in 1,000 bases is different as SNPs, there are these larger structural variants as well that cover many more bases. Okay, so how are these alleles distributed across the population? Now, this Hardy-Weinberg law comes up again and again, and deviations from it are really useful in terms of identifying genes that are under particular types of selection. So it has all these assumptions: large population, random matings, no appreciable rate of mutation. It turns out the rate of mutation is low enough that this is basically true, and no selection. So if you follow all these conditions, even for just one generation, then you end up with this Hardy-Weinberg equilibrium, where if P and Q are the allele frequencies, P squared is the frequency of homozygotes for P, Q squared is the frequency of homozygotes for Q in the population, and 2PQ is the heterozygote frequency. So just as an example, if you have 0.9 for P and 0.1 for Q, then that means 1% of the individuals are going to be QQ homozygotes, which is fairly high. 0.1 is not that rare of an allele, but it's very rare for even in that case to have Q homozygotes in the population. That's sort of the main point of Hardy-Weinberg equilibrium. Okay, so how can we take advantage of nucleotide diversity and DNA sequences to tell us which parts of the genome are important for biology? And there's different types of selection which give different signatures of in the genome. So one is purifying selection. Purifying selection is where you have a sequence where you're under pressure to maintain the sequence the way it is. So your typical coding gene would be under protein coding gene would be under purifying selection. You end up with reduced nucleotide diversity because mutations that occur that alter that amino acid sequence, for example, tend to be lost by selection, and so you have less polymorphisms in those sites than you would expect at a random site. The reverse of this is positive selection, and these are places where actually there's an active pressure to change the sequence. And so we talked about a couple examples. One of the classic examples would be lactase expression, right? So what happened with lactase is we had now probably a small number of mutations that actually caused the lactase gene to be expressed later in life. And that brought with it a number of genes that were nearby, and we'll talk about it in the next slide. But if you have an individual gene where the whole gene is under positive selection, you can actually find examples of this where you have more changes in the protein, for example, a protein coding sequence than you would expect by chance, and so that's a really strong signature of positive selection. And then there's been a number of recent studies looking at species-specific loss, so actually looking at genes or mostly regulatory elements which have been conserved over most of mammalian evolution, but then have been lost in human evolution, and there's speculation that those may be involved in relieving some kind of gene expression regulation that then allows human-specific traits to develop. So just looking at the diversity around sites of selection. So here's this is an example of a positive selection where you have a new mutation that gives a selective advantage. What happens? So this is the ancestral chromosome. You had one chromosome that had a mutation and that's the red asterisk, and there's lots of other DNA polymorphisms along the length of that chromosome that aren't present on the other chromosomes in the population. So after one generation, this has been selected on. There's more of this red chromosome, but it's also been recombination, so it's been broken down. So you have these breaks, and so the chromosomes that have your red mutation have a number of these passenger mutations around as well, but not across the whole chromosome. And after many generations, now the red marker has gone to fixation in the population. Right, so everybody has this beneficial red allele, but everybody also has a certain amount of sequence that was on either side of that red allele, just because there hasn't happened, by chance, there hasn't been recombination up to a certain point, and the distance between your mutation and the edge of the closest recombination is going to depend on how quickly it went to fixation, among other features. And so you have this block of new alleles that have come along with your selected allele. And that's related to a sort of a parallel concept, which we're going to talk a lot more about in the next lecture, which is linkage disequilibrium. So linkage disequilibrium is just basically saying that a number of alleles that are closely linked to each other are not randomly associated in the population. And the same basic principle applies here. So now you have some ancestral chromosome, and over time there's lots of recombination that's happening and breaking apart these individual variants. But even after a large number of generations, if you have a number of markers that are sufficiently close to each other, they will stay on the same haplotype, so they stay on the same individual chromosome as each other. And how big of a region that happens over, again, depends on how old those alleles are. So very, very old alleles tend to have small regions of linkage disequilibrium around them, whereas very young alleles will have much larger regions of linkage disequilibrium around them. And the advantage of this is that for common traits where we think that there might be some old rare variation that may impact the risk of the common trait, we can actually use this linkage disequilibrium. Basically, instead of having to sequence the whole genome, we can just sequence these individual SNPs, and that gives us sort of a surrogate for that entire linkage disequilibrium block. And we'll talk, like I said, we'll talk about that more in the next lecture. Right. Okay, so the last question. We have a sea squirt, Siona savini, which has a population size about 100 times larger than the human population, but this mutation rate is similar. So actually, Siona population size is about 1.5 million is the effective population size, which is huge. So what fraction of the genome would you predict is going to be heterozygous for sequence variants in Siona individuals? And the answer is actually, just from what I told you, should be about 1 in 10. If humans are 1 in 1,000, and I told you that the heterozygosity is proportional to the effective population size, then since the Siona population size, effective population size is 100-fold higher, then its heterozygosity should also be 100-fold higher. That brings us to about 1 in 10. And in fact, that actually was a big problem when they sequenced the Siona genome. It was hard to assemble it into a single genome because they sequenced it, diploid, and so you had basically separate assemblies for the two different alleles because there's so much variation between them. And the actual heterozygosity is about 5% for SNPs, but 16% for indels. And just point out that the reverse is true for endangered species. So if you have a population size of 20, then you're going to have extremely low levels of diversity. Right, and that's the end of this lecture. We'll talk next time about clinical genomics, and I just want to thank all these people and at Penn and online for help with images and slides.